Hi, everyone. My name is Patrick Hahn. I'm a second-year MBA student here at Tepper. I'm here with my colleague, Tyler Gilcrest, and we're here to represent Orbitz, and our mission is to accelerate preclinical drug development. So this company was founded by my friend Abraham Lin, who's a PhD at the University of Antwerp. He is an expert in organoid technology, and we'll get a little bit into what organoids are in a moment. And he's joined with a team of 3 other PhDs who are expert in organoids, wet lab development, and drug development, as well as AI and machine learning technology to use computer vision for accelerated drug development. Tyler is the commercial lead for this project, and we're really excited about the diverse talent that we have, and it's an honor to be here. So in terms of the problem we're trying to solve, you all are probably very familiar with this process. This is a typical drug development value chain. And as you can see, it's a funnel that goes from over 10,000 candidates down to a few candidates. And these pharmaceutical companies are looking for blockbuster drugs, right, that are going to be worth over $1 billion, right? But the main problem that we see here is that 90% of drug candidates fail. And why is that? That's because almost half of drug candidates, they lack clinical efficacy when they're being studied. And part of the problem is that we are currently using mostly 2D cell cultures and animal models, which can be very cost effective, but they're not as clinically relevant, or if they do have clinical relevance, they're not very high throughput or cost effective. So as you can see in this 2x2, 2D cell cultures, right, very cost-effective and efficient, but very low clinical relevance versus mice, maybe more clinical relevance, but not very high throughput, right? And this is where 3D cell lines and organoids comes in. They have the best of both worlds. You all may have heard about organs on a chip, which is gets at a similar value proposition, but organoids provide even more attractive cost structures, and it's just a lot more cost effective. The problem, though, with organoids is that organoid analysis is very cutting edge, is at the forefront of scientific development, and so it's very difficult to do, and not most people don't have this expertise. There's a lot of unique challenges with image analysis with 3D cell lines, and so this is where Orbitz comes in.
Thanks, Patrick. So what Orbitz is offering is a platform to go from the end of the microscope out to all of your sort of automated data analysis needs. And so the team in Belgium has developed, they ran into this problem themselves. Christoph, one of the PhDs in the team, organoid specialist, won a microscope, one of the first advanced microscopes of its kind in Europe, and ran into this problem where he was dealing with the issues of all the data that was coming out, the complexity, exporting things into Excel spreadsheets, manually manipulating things, and really running, bumping his head against these pain points. And got together with Abraham, Maxime, and the others on the team to build out AI tooling that can identify and characterize the growth of organoids and their reaction to therapeutic candidates in vitro using organoids, which are great patient models that really represent the environment that you get in the patient rather in the in vitro setting. So the features here, we have cloud data management in terms of compression. We have AI-based image analysis and automatic data visualizations. But really what that translates into is faster analysis, cheaper time in terms of the amount of time you're spending doing all of that analysis, and then ultimately making better decisions because you're using organoids in your studies of the therapeutics that you're screening. And so we see Orbit's value proposition in 2 places. On the left here, is what we're focusing on in the current present day, which is drug screening, getting better candidates into the clinic, making sure that we're confident about the candidates that are being accelerated into clinical trials, and when we're committing to those big timelines and budgets that go into developing drugs. And then on the right-hand side, this is our future vision of being able to use our analysis platform in more of a real-time patient treatment setting. So we're able to derive organoids out of patients' tumors and their specific tissues, experiment with therapies that are approved for that disease, but really tailor and customize the therapy to the patient, because we're able to run this analysis quickly on organoids that are derived directly from that specific patient. And so that's much further down the line, but where we want to go in the future with our analysis platform. And if this wasn't a PDF, you would see a cool GIF of our technology there working on the left-hand side. But if you can imagine, we're able to characterize the growth and the state of the organoids over the course of the urinalysis. And so we're able to look at both the organoids on an individual level and whether they're growing, whether they're dying, but also on a population level, and analyze each individual well to identify anomalies in the well, if it may be different from other wells that you have in this high throughput screening setting. And then on the right-hand side here, we have a small clinical trial of patients with the University of Antwerp Hospital that showed that our analysis was able to predict patient response to therapeutics ex post facto. So this was taken from, these are organoids that were derived from patients who were then treated with therapies, and were able to predict whether the patient would have responded to those therapies based on whether they did after the fact, which is great early data. It's very small. It's in our lab specifically, but it's a positive indication that our analysis is trending towards where we want to go in the future with that sort of very personalized medicine. And so I'll just close out kind of with a case study of what we've started doing so far. This is Hendrik. He is a researcher at a group called IMTM in the Czech Republic. And he has a workflow where he needs to screen thousands of compounds. They have a biobank, a database of over 100,000 compounds that they're looking for drugs for repurposing purposes. And his workflow is applying masked images, exporting things to Excel, checking for errors, and doing this iteratively. So up to the point that he was doing this for 19 days. But when we did that exact same workflow and produced some automated reports for him as a result of that, we were able to do that same process in less than 24 hours. So what we're hoping to do is really make the drug screening portion, a lot more efficient for these companies that are developing these drugs, and really bring these drugs faster to market in the current day, and then move into that personalized medicine into the future. So thank you. We appreciate the opportunity to speak here. So thank you to the organizers. And if you have anyone that is doing organoid work, or even high throughput screening work in general, connections that we could talk to, learn from. We're still very much doing customer discovery, trying to nail down where our value proposition lies and who benefits the most from this. We'd love to chat. Our contact information is here, and we'll be around at the event as well. Thank you.